Ireland has a long history of conducting national food consumption surveys. Having carried out the first in 1946 to assess food intake after the war, several large-scale studies followed, collecting detailed information on 8,800 adults, teenagers and children, an investment of almost €8.5 million. These detailed national surveys have allowed us to take a snapshot in time of what the population is doing. They have provided an invaluable resource, allowing us to assess trends in health and behaviour, guiding legislation on the safety of our food, and developing policy and regulations to prevent disease. However, as national surveys typically measure hundreds or even thousands of people at any one time, they're expensive and therefore usually limited by the amount of data they can collect, typically consisting of a dietary assessment, fasting blood biochemistry, basic body composition, and some measure of physical activity. Together, these measurements that describe our body characteristics are known as the person's phenotype. If you look in the dictionary, however, phenotype refers to any observable structure, function or behaviour within a living organism. In other words, just about everything measurable within the human body. Therefore, there's potentially a lot more which we could see. This need to study the totality of human biology has long been recognised by researchers, nutritionists in particular, and the concept of the nutritional phenotype was born well over a decade ago. The concept behind nutritional phenotyping is to bring standard dietary databases one step further, to combine these traditional measures with newer technology-driven sciences known as nutrigenomics or omics technologies. While omics technologies are most widely used in the scientific literature to refer to the science of genomics, the study of analysing the genome or entire DNA sequence, other omics sciences exist. Transcriptomics, for example, allows us to analyse the process by which small segments of our DNA are transcribed or copied into RNA, an essential first step in assessing the activity or expression of our genes. Proteomics allows not just one but all measurable proteins within a biological sample to be analysed at once. And metabolomics provides the potential to test thousands of small molecules which are the product of our metabolism. This combining of the old and new in nutritional phenotyping therefore has the potential to more completely assess the impact our diet has on our metabolism. In 2007, the Irish Government Department of Agriculture, Food and the Marine, recognising the importance of this emerging area of research, proposed fundings for research projects in the area of nutritional phenotyping as part of its Food and Health Research Initiative. Experts from the nutrition departments of 4 Irish universities, University College Dublin, University College Cork, Trinity College Dublin and University of Ulster Coleraine, with researchers from several Irish hospitals, came together to submit an ambitious research proposal. The plan was to create an Island of Ireland consortium on nutrigenomics, called the Joint Irish Nutrigenomics Organisation, or JINGO for short, and for this consortium to create a national nutritional phenotype database. The proposal was successful and received €5 million in funding from the Irish government. In 2009, it was to go on to receive a further €1.5 million from the Northern Ireland Department of Employment and Learning. This large project collectively became known as the JINGO project. One of the pioneers in the area of nutritional phenotyping and lead investigator of the JINGO project, Professor Mike Gibney, speaks now about JINGO and the future of the nutritional phenotype.
The first thing we did was to set up a cohort which was nationally representative, which we call NANS, the National Adult Nutrition Survey, which is based on 1,500 people with a fasting blood sample, diet and many other variables measured. That was the first cohort. Now, the problem about fasting blood samples, they say, is that metabolism is asleep at the fasting level and that it only becomes awake when you've had a meal. And then you begin to see enormous differences between individuals, which you don't see in the fasting state. So we wanted a metabolically challenged group, which we nicknamed MESH, and we had just over 200 people that came in on 3 different occasions. The first 2 occasions involved test meals, either a glucose meal followed a couple of days later or weeks later by another glucose meal, fat followed by fat, and then all groups came into the sports laboratory for detailed physiological exercises and detailed imaging. So that gave us a database which really challenged people with a metabolic challenge of a meal. Now, the third group we looked at were people who were metabolically challenged not because of a meal, but because of their age, because aging brings with it a decline in the function of a number of physiological processes. And we wanted to study how that in that particular group, nutrition related to genes, related to phenotype. So we had 3 groups within that. We had around about 1,500 people, you could say, in each group. One group had hypertension, one group had poor cognitive function, and one group had impaired bone function. And they were all clinically established. So we had 3 very different but very contrasting and useful cohorts. In my view, the JINGO project is pretty unique, and I don't see too many projects around the world which have the same level of coverage that we have. I mean, I've described the 3 cohorts we have, but in addition to that, what makes us special is the extent to which we burrowed down deep into these people. We measured gene sequence, gene expression, we looked at metabolomics, we looked at lipidomics, we looked at proteomics, we had particular plasma profiles for bone disease, for folic acid metabolism, for cognitive function. We had people that were with detailed physiological functions. We had detailed imaging using DEXA scans, and that makes it pretty well unique. Since we began in 2007, I think the biggest change has been the global interest in having large-scale nutritional phenotype databases, and a recognition that what we've been doing in the past, which served us very well in the past, with the new dawn of biological capabilities, we need to go a step further. So I think the big change is the realization among many funding agencies that they need to engage in large surveys, and indeed to pool large surveys. And I would say the recognition of the need to pool surveys is the biggest change that has come about since we started in 2007. In my view, the big direction that we need to go for nutritional phenotype databasing is standardization. We know a lot about carrying out dietary surveys and the methodologies that should be employed. When it comes to metabolically challenged meals, we really do need to standardize exactly how this is done. We need to standardize the caloric intake at the test meal, the nutritional composition of the meals we feed. We need to have standardization of the time of blood sampling for different types of meals. And we need to understand how to control, if you like, the baseline of the diet the person leads into having a test meal. So those are areas which are very difficult to study at the moment, but we will have to sit down and work out experiments to see how best to develop the test meal.
The Member States of the European Union, in association with the European Commission, have created a joint programme initiative called A Healthy Diet for a Healthy Life, and this international initiative has identified a number of activities that require coordination and funding across the Member States, one of which is the need for a European-wide nutritional phenotype database sharing initiative. In 2013, in line with this, the Irish Department of Agriculture, Food and the Marine issued an open call for Irish-led research proposals, one of the themes of which was food for health. The JINGO consortium submitted a proposal in May 2013, and the project was approved and awarded funding in November of that year. This 2-year project, which will run until the end of 2015, is called the European Nutritional Phenotype Database Sharing Initiative within the Joint Programme Initiative, or JINGO JPI for short. Over the course of the 2-year programme, JINGO JPI will see a significant amount of nutritional phenotype data being uploaded to a central European repository set up by the European Nutrigenomics Organisation, NUGO, named the DBMP. Data will come from all 3 JINGO databases, as well as from 2 other studies, the European FP6 project LipGene, and a new study called the UCD Twin Study. In total, information from almost 7,500 adults of all ages.